Welcome, everyone, and thank you for joining us at today's seminar, Organs on a Chip, a Revolution in Drug Development, presented by Dr. Gorm Yoder, a Scientific Director in Strategic Science Excellence and External Innovation at the Janssen Pharmaceutical Companies of Johnson & Johnson. My name is Dr. Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing the presentation, just click on that Ask a Question box and let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process for obtaining those credits. Now, without further ado, please join me in welcoming our presenter today, Dr. Gorm Yoder. Welcome, sir.
Thank you. I'm very happy to be able to speak to you today about technology that I've been looking at for some time. And what I want to do is basically give you a description of that technology and hopefully some insights on where that technology can be applied. And so basically what I'll do is give you a brief introduction about my role at Janssen. So part of that role is looking at innovation and looking at new technologies within analytical development and for the small molecule organization, which I'm a part of. And then I'll get into a more general description of microfluidics technology, which is really, in my mind, a driver for a lot of technologies that we are beginning to see and will continue to see in the analytical space. And then, of course, organs on a chip, which is really the main topic of today, which is basically microfluidics technology, but then combining elements of biology to that. And then I'll get into the problem statements for biotech and pharma, and then discuss a little bit about where we are today, the next steps, and then some conclusions. So first, what I'll do is I'll describe a little bit about, or give you some thoughts about science and innovation. And so, science is mostly about evolution, but revolutions do occur. And certainly within my career, we've seen a lot of innovation. We've seen a lot of things happen. But I would say for the most part, things are very incremental. Things are gradually improving. New technology introduces itself, and it's very much of a continuum. But in some cases, you do see some startling changes and things that do occur. In terms of how we view innovation, we typically look in a longer term, 10-year horizon. And in order to do that, what you have to do is visualize the end state. Where do you want to be in the next 10 years? And so obviously that's a difficult question for a lot of people to answer, but it has a lot of relevance, I think, if you're certainly working in pharma in terms of thinking about what are the needs for, the medicines of tomorrow and some of the new modalities that we continue to be confronted with. And so oftentimes in this discussion, then we think about creating a roadmap or, sort of a means to get there. And so what I've sort of put in on this slide, it's really kind of more for fun, just to kind of introduce you to the concept. But, and certainly, most of you have probably studied a little bit of philosophy and physics and chemistry, and there's been a continual evolution that's occurred. And to me, what's striking is that as we sort of incrementally move forward, there's always this accumulation of collected facts and information. And usually what happens is that you get, a series of anomalies which occur, which then sort of at some point, when they reach a critical mass, it gets us into sort of a crises mode where maybe new theories have to be developed. And so, and oftentimes I think it's hard for us, scientists to let go of, things that we learn in school and have applied our whole careers. And so I guess the way I look at innovation is sort of letting go of certain preconceived notions and trying to look forward and trying to determine where I want to be without really thinking about, okay, what are the challenges or what are the impediments? Just thinking about what is the future state going to look like, and really thinking out of the box and trying to, move forward. So, in terms of revolutions, I mean, I think already we're experiencing one, and I think most people will probably agree with this. Is that the biggest revolution going on right now is in data sciences, and so. And it's particularly felt, I think, when you sort of look at sort of the Generation Xers and the Millennials, you see this sort of transition occurring where where I like to call it going from the analog to digital world. Suddenly you have the internet, you have social media, and then we're starting to use tools of artificial intelligence and machine learning. These are things now that Facebook and Google and Microsoft and Apple are using. Amazon, they're using this on a routine basis. They're generating copious amounts of information and using that and trying to predict outcomes. And so, kind of reflecting on this, I would say 20 years ago, and I've been in the industry for a while. Much of the technology we see today was, maybe considered science fiction. I like to kind of call these new things science fiction. But what's happening more and more is that we're seeing technological advances really accelerating. I mean, it's really pretty incredible now. What the digital age is really bringing in terms of informatics and information and how we process that information. And so really, I think when you're looking at innovation and looking at just how scientists need to adapt, you're really confronted with really one decision. Either you go with it, or you get out of your comfort zone, or you don't. But in the end, I think you have to be bold. So. So why do I want to preface this with microfluidics technology? Well, I think I sort of preface the discussion with informatics and data, and so really a lot of the expectations right now in science is that we generate data quicker, more efficiently, and so microfluidics basically is a technology which can help enable that. And you're really seeing this start to assert itself into the research laboratories, and what it basically deals with is being able to precisely control behavior and manipulate fluids that are really constrained to a small scale. And so basically, if you look at, let's say, typical analytical instrumentation, what we're doing is really miniaturizing it in many cases to quite a miniature scale, or a chip, as we might call it. And so this affords us the opportunity really to integrate one or more different analytical functions or protocols within a single circuit. And so this is really quite amazing. And so organs on a chip, it's really kind of a logical successor to this, and I'll get into the the specifics around organs on a chip in a little bit later in the presentation. But typical advantages of microfluidics include the miniaturization of standard laboratory tools, low fluid volume consumption, which means less cost and waste, high throughput, which as I mentioned is an important consideration, so that basically translates into faster analysis and response times due to shorter diffusion distances and things like that, better process control, and finally, it also is a potential enabler for medical diagnostics, wearables, and biomarkers. So what I'm going to do for the next few minutes then is really kind of give you an introduction of what organs on a chip are. And again, I want to preface this discussion with the fact that I'm an analytical chemist. I'm not a biologist, but and so maybe folks that are listening to this presentation will hopefully be able to explain maybe some of the intimate details a little bit better than I can in terms of the biology. But what I want to do is just give a high-level overview and where I see the potential of this technology going. And so organs on a chip, or as we sometimes call microphysiological systems, are basically microfluidics devices in which one is able to or attempts to basically put cell culture and simulate physiological activities, mechanics, and things that occur in various organs or organ systems. And so, as I had mentioned earlier, this is really about the convergence of lab on chips with cell biology, which has a lot of potential to permit the study of human physiology in an organ-specific context, and introducing novel models for in vitro multicellular human organisms. And so one can see a potential of being able to look at various organ models simultaneously. And what I have listed here are a few of them that are actually being actively studied in this particular arena. And so you can look at things like lung models, which focus on pulmonary inflammation or infections, liver toxicity. Again, tox is a major concern in drug development and discovery, and so having the ability to look at screen different molecules for toxicity is extremely important. Also, there's mechanisms around the blood-brain barrier, which is a crucial biological interface, and really has to do with drug transport, delivery, and of course drug safety. Then there's also models around the gastrointestinal, the GI tract, which can emulate gut biology, disease, and toxicity. And then we can also look at skin effects. So looking at corrosion, irritation, sensitization, photosensitivity, genotoxicity, et cetera. And finally, cardiovascular. And so there's, any number of different physiological systems which could potentially be evaluated and are being evaluated. And again, this is a short list that that I've put here, but there's obviously other ones that potentially could be evaluated and assessed as well. So, giving a little bit of context, so this isn't something that is sort of off of everybody's radar. It's actually starting to get a lot of funding now. And in around 2010, the NIH and the FDA co-founded the Advanced Regulatory Sciences Initiative, in which, and then DARPA, which is the Defense Advanced Research Projects Agency, shortly thereafter joined in. And basically, the focus was really on how to, evolve this technology even further. And I'll get into the why, we would want to do this, or where I see the potential of this going. But recently, there's been quite a few awards totaling around $15 million per year to develop these microphysiological systems and platforms to model human disease. Now, I might add that there's a lot of different potential platforms out there, and I think that's the challenge right now. And certainly with new technology, there's always a risk that if you invest, it may not be around tomorrow. So I think that's also, a bit of a challenge. But I think there's some ways to go about that as well, which I'll get into a little bit later into the discussion. and then, and as I'd mentioned before, potentially multiple organ models can be evaluated simultaneously. And so where I see this technology going is really building, eventually building an end-to-end platform for discovery and development. So we start basically in looking at different targets. So in drug development, often, we're looking at different molecules and trying to figure out which ones, meet the criteria or best meet the criteria to move on and introduce into the various clinical phases. Of course, toxicity is a major concern, so we want to make sure, that's the number one concern. We want to make sure that there aren't any safety concerns to patients by taking any of these medications. And then, of course, once you've chosen a molecular entity to develop, then you get into really looking at, okay, what is the clinical efficacy, the dosing and things like that, biomarkers, and, the bioavailability. And so I think, at least for me, where I see this is, if this technology, if we can learn from this technology, or get more, learn more intimate details about human physiology via this technology. And if we can maintain these models, then I think, this has a lot of potential, especially during the life cycle of a product where maybe we look at different formulations, we look at different dosing regimes and things like that. So I think this has, again, really a lot of potential. So, as far as where we are with this technology, I mean, I think we're kind of at the early stages of thinking about complex physiology. Now, I think there's a lot of companies out there, and I won't get into the companies that are actually involved because I don't want to sort of give the impression one is better than the other, but certainly if folks are interested, we can talk offline about this. But I think in general, there's a lot of different platforms being developed and different approaches to this technology, which involves multiple interacting cell types with an organ. And I think, certainly one of the challenges is that the complexity often requires a trade-off in throughput. And so, with the, essentially what you're looking at are microfluidic cells that need to be controlled. Different parameters need to be controlled simultaneously and monitored. So things like media, flow, temperature, et cetera. And so really, if the more questions that you're trying to answer, I think, the more impact it has on the throughput, and certainly it tends to slow things down. Now, I think there's a way to circumvent this as well, and there are people that are looking at this from a different perspective and maybe looking at multiple chips simultaneously as opposed to just a single chip. And then I think one of the major challenges is also the cell sourcing of the primary cells, from human tissue, and those can be difficult to obtain. And of course, you want to model around and be able to differentiate between healthy and disease states. And so, being able to get access to stem cells is obviously a challenge. But I think most importantly, I think consistent sourcing is really difficult. As I'd mentioned, there's a number of players in this space right now, and the list is ever expanding, which are trying to emulate a wide range of tissues and organ systems that oftentimes, employ different approaches. And so what we'll eventually have to see in the end is some level of standardization or platforming. I mean, it's impossible that we have, let's say hundreds of different approaches. But again, I think certainly where the focus will be is a little more, let's say, agnostic to the specific technology that's being provided by a different vendor, but more on the approaches and how we validate these approaches and these models. And I'm going to get into that a little later in the presentation. And so in the end, I think what's really needed is really a symbiotic environment where you have a feedback loop that's created between research institutions, biotechnology, and pharma. health authorities as well, to for this technology to evolve and eventually reach widespread applications. And again, I think the focus will be on platforms that, platform agnostic approaches. And so again, if we take a technology, how do we validate that? How do we show that it's robust? How do we show that it's reliable? I think that in the end is going to be, the ultimate arbiter. And, in addition to the question that is being asked of the technology. So, what I want to do is get into some of the intrinsic challenges encountered in drug discovery and development, and why we even want to consider this technology in the first place. As most of you probably know, at least that work in the industry, pharmaceutical research and development efforts are often both inefficient and suffer from a high attrition rate of drug candidates within the clinical phase of development. And this number, 95%, maybe varies a little bit from company to company, but if you really look at the failure rate's quite high. And so, the number of drugs that actually come to market is a representation of many failures as well. So, so obviously there's a driver to increase the odds. And what this basically, is indicating is that, we don't know enough about the human biology and the human physiology. And so, being able to use technology such as organ on a chip, and of course there's others that maybe are being considered, but again, I think, the rationale for pursuing this type of technology, again, is addressing the very specific problem of the high attrition rate of drug that occurs in drug development. And what we see is that most drug trials either fail to meet their expected endpoints or show minimal statistical differentiation between active and placebo. So that really speaks to the efficaciousness, or they're just not, let's say, significantly better than what's out there as well, which is now an important criteria. And then certainly there's other instances where failures may happen for safety reasons, and usually, the tox studies, we do tests in animals, and again, I think this is also another potentially another means of avoiding maybe animal testing and being able to get some immediate reads on tox, which wouldn't otherwise occur through traditional means. And so, again, as I've indicated, this is clearly a fundamental. There's clearly a fundamental understanding of need to understand drug properties in drug development and the early phases of development, as well as accruing knowledge regarding the specific mechanisms of disease. And so, I think the data management piece that I kind of spoke to at the beginning of the talk is really important in this as well. Drug companies are generating a lot of data, and the data that's generated, both successful data and failed studies, I think has equal relevance in terms of learning and understanding. And so I think in the end, the companies that are best able to manage the data are going to have a significant advantage over the other companies. So, maybe that's a separate discussion for another time. But again, I think the revolution that's occurring in data sciences, which I sort of preface this talk, is real. And I know certainly within Janssen, we're trying to make efforts to address it. So in terms of the future state, clearly we want to look at alternative approaches and technologies to increase the productivity and success of ongoing drug development efforts. It's not good for us, and it's not good for the patients either. So certainly, I think at the end, if we can bring more drugs to patients and more therapies to patients, that's a win-win situation for everybody. So, assuming that much of the prior knowledge from drug development and trial efforts could be aggregated in some form of management system, It's been proposed that systematic modeling and simulation-based trial designs could enhance the odds for successful commercialization of new drug entities. And to that end, the application of microphysiological systems or organs on chips for development and discovery, which can enhance, the enhanced in vitro models can be then used to stimulate and predict in vivo processes. And all this is starting to show a lot of potential right now. It has a ways to go, but certainly it's starting to show a lot of potential. So, assuming that reliable in vitro models could be developed, I think there's several valuable outcomes that can immediately be visualized. So the obvious ones are enhanced mechanistic understanding of disease states. And then, of course, as I alluded to earlier, the improved and more efficient selection of drug candidates. And then I think what's really important as well is the decreased reliance on animal testing and a more humane framework for animal research. And of course, improved clinical outcomes. And then the most important, I think, of all really is more therapeutic options for patients. And so, while the utilization of MPS-based technologies could someday reap enormous scientific and regulatory benefits for patients, there's still certain barriers that must be overcome, which are impactful to the evolution and future implementation of these technologies. So in terms of next steps, I've listed here some assumptions. And so I think some important assumptions include more careful exploration of drug properties early in development is critical. So really we're speaking about the developability. And so, again, I think some of these technologies, I think, could potentially help us here. And then to allow for more efficient planning during the early phases of drug development. There's a need for the accrual of disease and trial knowledge from across various development programs. So again, the data management topic is extremely important. What have we learned from the past, and how can we leverage that to help us in the future? So the data management then enables agility, efficiency, and proactive decision making in the end, and helps us make better choices about the active moieties and molecules that we choose to pursue and develop into hopefully commercial drugs. So in terms of modeling, I mean, there's already a lot of modeling that does occur, and the modeling itself is certainly something that's recommended by the FDA and is a potentially valuable tool to improve the predictability of and productivity in the drug development process. And so, there's a few different approaches here. So there's the system biology approach, which, basically you're mathematically representing the biological system at the molecular level and overlaying that with pathological perturbations. And so these are what we call physiologically based models. And this is kind of where the organ on a chip sort of resides. And so looking at, can we simulate it, in vivo activities and create an in vitro model through, the use of those chips. And then there's the semi-mechanistic and the empirical models. I think the semi-mechanistic tries to simplify some of the biological, simplify the biology enough to describe what the data is telling you. And then, of course, there's the empirical models that really are just data driven, and they just kind of tell you, well, it either works or it doesn't, but it doesn't really have as much, it doesn't have any strong attachment to the biology, so to speak. And so one of the important aspects of the systems biology model is the characterization of the relationship between the biomarkers and the clinical outcomes. And so I think this certainly is something that is already being studied, but I think, again, technologies that can allow us to maybe look at this in a more high throughput sort of perspective and be able to allow us to screen more agents, I think is going to be extremely helpful. And so, within the microfluidic construct, there's various bioprocesses, again, that are simulated, and the outcomes from multiple experiments are then overlaid with mathematical models that estimate or predict in vivo performance. And then these mathematical parameters then can be continually refined as either existing data is obtained or reassessed or becomes available. So the regulatory configuration. So I think, certainly once you've decided to sort of jump into the ocean of organs on a chip, I think what you'll find beyond just the, once you get beyond just the science of it, there's certainly an important regulatory consideration that needs to be discussed and developed. And so that narrative, I think, is probably still, I think it's, it still has a ways to go. And again, I think, any new technology that you are introducing ultimately has to be understood by certainly the regulatory authorities and the reviewers. And also it's also held up and compared against, orthogonal methods or existing technologies that are, let's say, readily available or widely accepted as standard tools. And so, again, there's always a little bit of a threshold that you have to overcome in order to introduce a new technology. Now, it's not to say that it's impossible, and it certainly isn't. But again, I think you have to do a lot more, let's say, convincing in some ways in the beginning, and educating, I think, is the most important aspect. Being able to educate the authorities on how this is applied and why this is meaningful. So, owing to the fact that these are new and evolving technologies, there's really no regulations that exist for use of organs on a chip in drug testing. So, toxicity and PK testing require the use of specific animal models for specific periods of time, which requires that the technology be sufficiently evolved and understood to displace these traditional practices. So considering that organs on a chip are a type of analytical instrument for the purpose of drug testing, I think there's several current regulations that could be applied to their development and implementation. And so things like chronic toxicity testing requires compliance with good laboratory practice regulations, which includes equipment calibration, validation, and qualification. Commercial drug identification and assay test instruments must also comply with ICH-Q2, which is basically a guideline that describes validation of analytical procedures. And then I think even in this context, I think traditional validation parameters, such as accuracy and precision, while they may not apply specifically to this technology, we still have to show that that we can create models that are sufficiently robust. So, again, validation in the classical sense for an analytical method may not follow the same trajectory for organ on a chip. So, again, while validation parameters such as accuracy and precision may not be applicable for MPS systems, validation protocol design and results should be geared towards demonstrating reproducible results. So, you want to make sure that each time you use this technology, with certain agents and things like that, that you get reproducible results. And then I think what sometimes people forget, and I think this kind of goes into certainly late development and commercial, is the maintenance of these models. And I think, potentially they can be a very strong tool to address questions that oftentimes come up during the life cycle of products. So to sort of give you a quick example, oftentimes we're changing vendors for materials, and things can happen. Things can sometimes the chemical and physical properties can change slightly, and we need to understand that. And need to understand whether that's it's an important outcome. And so again, I think if we have models that we can leverage that can tell us if there's a real significant or important in vivo impact, I think that's important. The FDA runs, voluntary qualification programs for biomarkers, clinical outcome assessments, and animal models. And so, the program helps sponsor and develop such tools for the purposes of drug development. So the formal process involves consultation with the FDA and a review with qualification procedures for the development tool. And of course, at least for me, it goes without saying that organs on a chip can be a useful addition to this program, which helps companies implement the technology during the early phases of development. So, the modelings, the in vitro models, the real purpose is it's really intended to handle future risk evaluation. And so alternative methods need to fulfill, sort of the fit for purpose criteria for regulatory acceptance. And so the EMA, which is the European Medicines Agency, basically gave out 3 criteria. So a method needs to be valid, meaning it has standardized protocol and clear readouts. And further, it needs to be reliable, relevant, and validated by recognized institutions. Second, the method should generate new and meaningful data or replace an existing method with the same or better results. So again, you're always going to be up with, sort of proving that your method is at least as good as the existing one, or hopefully even better. And some of this I think has probably has some relevance to standard business practices of various companies, and so it can be a different discussion depending on the company you're working with. And then finally, what's expected is that the new method must have been applied in parallel but independently to an accepted method in a regulatory decision-making process. So again, you have to, with a new technology, you have to certainly compare it against orthogonal methods or methods which are standard for the industry. And so in order to achieve these criteria, then the qualification of the equipment operating the device according to the standard installation, operating, and performance qualification is required. The generation of reliable, reproducible, and robust data needs to be assured by adherence to rules of good cell culture practice. So again, organs on a chip, we're talking about tissue and things like that. So the use of qualified cells and tissue sources is extremely important. And again, I think that's sort of an intrinsic challenge at this point with the implementation of this technology. But again, that's something that will, I think, improve over time. And then the improvement of existing models and assays towards closer recapitulation and in vivo physiology has also been recommended. So, certainly the closer we can get to predicting physiology, the better. And so, so defining, I think, then the critical regulatory gaps which can be addressed by MPS is essential to define the biological and functional features of the model system. So really, also, highlighting what is the question that's being addressed, and certainly there's always gaps that we encounter in development, and so I think certainly if you're going to introduce a new technology, the thought is that you're hopefully addressing a gap that couldn't otherwise be answered through standard practices. And then I think certainly in the application of these technologies, there's an expectation that a library of reference compounds for qualification would be used. So, there are certain compounds that would be specific to certain disease states and things like that. And so that's something that I think is an expectation that, if you're going to introduce a technology or your version of this technology, that it's it can at least be referenced and compared against known compounds that are well characterized and well understood. And then, of course, again, which goes sort of without saying, is that the scientific basis needs to be properly described. So this, so with the technology that you're using or the type of model that you're trying to introduce into your development or discovery, this includes information around the biological and physiological processes that are being modeled, and why they're thought to be relevant, for the substrate's adverse effect. So, and so finally, then the validation roadmap. So if you look at how this would sort of evolve and where we would end up being, I think there's a couple of different sort of outcomes that potentially could occur here. So the first one, basically, systems aiming for safety and efficacy testing during drug development might be qualified through the U.S. FDA drug development tool, so the DDT qualification program. And this is especially true for systems requiring fast track validation, such as those MPS models targeting compounds of diseases that cannot be tested using techniques currently available. And so this applies especially to novel biopharmaceuticals, specially designed for humans, and maybe those that are irrelevant to testing in animals to begin with. And so here, a clear description of the manner and the purpose and the use of the systems, as well as its limitations, is really mandatory. And then the validation, I think I've kind of alluded to this, validation of individual biomarkers under the biomarker qualification program on a specific MPS may be a key step to our qualification acceptance as a tool for therapeutic development as well. And then finally, qualifying an MPS for specific context or use as a drug development tool will eventually lead to a tool which may be considered product independent and may be used for efficacy testing and development programs for multiple investigational drugs for the same disease or condition that's being targeted. So, additionally, I think I kind of alluded to earlier in the talk to an important consideration about replacing, or the aim to replace, animal testing with in vitro models. And so, so the MPS, that aim to do this need to be, at least oblige the respective OEC guidelines. So that's the Organization for Economic Cooperation and Development. And in the process of identifying that, hazard potential is highly standardized, and it's recommended that the MPS adhere to these guidelines. There's a guidance document on validation and international acceptance of new and updated test methods for hazard assessment. So, again, this is a critical criteria. And then high quality data from MPS that are consistent with the results derived from other approaches will provide initial evidence for the reliability. But again, a certain amount of care must be taken when comparing results gained from human-based MPS with animal studies, as the latter might lack, a certain level of sensitivity and specificity. So again, we're sort of in this conundrum of, we're comparing against existing tools, which may have their own limitations to begin with. And so again, how do you need to be able to differentiate between your tool and something which, may be lacking to begin with, which is not uncommon. And then, of course, doing then the cross-validation trials, to assess, the baselines would be constructive. And so, and then I think, the demonstration that these new devices can be used to identify compounds that have failed in clinical trials would generate more confidence in these technologies as well. So again, I mentioned, it's certainly success is important, but also failure. So I think if you really have a technology that can differentiate between both, or at least to a high level, be able to predict the outcomes, I think you have something that's extremely powerful. So some final remarks on this technology. We've covered a lot of ground, and so what I want to do on this final slide is really kind of highlight some of the, at least for me, the important elements of this technology, and sort of where we need to go, and sort of where we are at this point. So, as I'd mentioned, I think this is kind of more for the laboratory people, but the organ on a chip is really a successor to lab on a chip. So it's again, it's a microfluidics technology. So that means certain things, I think, to certain people, and certainly as an analytical chemist, to me, it means looking at it as an analytical tool and applying it as I might typically would, as an analytical chemist. And so, and I've certainly brought up some perspectives on validation, and what that might mean. And so, again, it's really the convergence of lab on a chip and cell biology, and so where I see this as having a lot of potential is if we can emulate human physiology and create in vitro models. We can really then directly address this high attrition rate that occurs in drug development, and ideally get more products to patients. And I think that's, for me, that's the number one goal. I mean, we spend a lot of time, a lot of people spend a lot of time doing a lot of good science, but it's a significant challenge. And, the physiology is extremely complex machine. And so, again, I think if we have technologies that can maybe give us some, a better read on what might happen, I think that's going to be a benefit to everybody. And as I'd mentioned already, these models are starting to show a lot of potential. I think certainly there's still some work to be done. But again, I think it's moving in the right direction. And certainly there's a lot of different valuable outcomes that can occur, obviously, beyond just the attrition rate that I've described earlier in the talk, but really getting an enhanced mechanistic understanding of disease states. I mean, I think certainly if we knew everything there was to know about disease, we wouldn't be, development wouldn't be so difficult, but it's challenging. And of course, then being able to quickly, I think what's equally important is really improving and being more efficient about the drug candidates that we choose to develop. So really looking at the developability, are these really fit for purpose? Are they going to meet the endpoint? And again, and certainly in pharma, we want to focus on the things that we think are going to work or have a high probability of working. So if we can quickly kind of separate out the bad apples, that would be a good outcome. And then I think, as I mentioned, decrease reliance on animal testing. I think that's important. And certainly a more humane way to go about doing clinical and pharmaceutical research. Of course, if we understand the disease states and the mechanisms behind the disease states better, then we can improve the clinical outcomes and then have more therapeutic options for patients. And I think, as I mentioned before, what's really critical in this is that we have a feedback loop between the research institutions that are really driving this technology and developing this technology, and with biotech and pharma and health authorities. Because again, a lot of times it's about framing, and certainly you have a lot of, let's say, really top-notch scientists that are working on this type of technology. But if you ask them, what are the typical questions that maybe we're confronted with in pharma, I think a lot of them would be hard-pressed to answer that question. I think it takes, certainly, you need to have experience in the industry and have gone through some of the challenges and trials and tribulations to really understand that intimately. So I think, again, really fine-tuning, the applications is critical, and that needs to really occur through dialogue between research institutions and the pharma industry, and of course the health authorities as well. In terms of how do we make this viable, and how do we show that it's reliable and that it's robust and that it's going to give us accurate and predictive information. And then certainly, at the end, there's, again, an absence of regulatory guidance around these, regulations. And so, of course, as this develops, I think there's going to have to be some guidance written. And I'm actually a member of the IQ Consortium. It's the Innovation and Quality Consortium, and there's an MPS affiliate which I'm involved in. And what we're trying to do in this regard is really sort of look at this technology agnostically, not really focus on specific vendors or, let's say, developers of this technology, but really look at, well, how would you apply this, and how can we make this work for everybody? So, again, I think those are things that need to be worked out. And finally, I think we get to the end of the conversation, but I think at the beginning of the presentation, I was asking if this was a revolution in drug development, and at least for me, I think the answer is yes, but we have a ways to go. So, as with anything worthwhile, even revolutions take time. So, thank you for your time.
And thank you, Dr. Yoder, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through May 2019. As a final reminder, Dr. Yoder will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us. We'll see you next time. Bye-bye.